sort of investigating the impact of tumor-induced cellular interactions and how that kind of plays into cancer progression within the body. So what is the tumor microenvironment? So when we look at a section or a cross-section of a tumor under the microscope, there's many more cells besides just your typical sort of tumor cells. There's a lot of vasculature, there's a lot of immune cells, and there's a lot of cells that kind of frame the bedding of the niche of the tumor. And all of these go hand in hand in contributing to tumor growth and proliferation within the body and are necessary for its survival. So if we want to take a look, we'll see this very complex overall structure of what the tumor microenvironment sort of has. It has within a lot of different immune cells, such as natural killer cells, dendritic, neutrophils, B and T cells. It has with it some of our other cells, such as mast and eosinophils, and that we will kind of go over throughout this presentation. And all of these basically go hand in hand and are necessary for the tumor to be able to get established within the body and then to evade all of the immune responses that the body would have and to just go and proliferate. So a very, very famous theory in cancer research is called the seed and soil hypothesis, and it basically talks about how the right seed can't really ever grow if it doesn't have the right soil. So no tumor cell on its own has the capacity to become a full-on blown tumor without the microenvironment now assisting and contributing to its development. And so now we're going to look at this sort of interplay and how we can study that in the context of liquid biopsy. So the cell types that we would find in the tumor microenvironment are, this very, very big heterogeneous group, and they're comprised of many different things. But today we're going to focus on the three main cell types, such as fibroblasts and cancer-associated fibroblasts, endothelial cells undergoing EMT, which is a phrase that you might have heard a lot, I'm sure, and then some of the immune cells, both from the innate immunity and the adaptive immunity. So first of all, we're going to focus on fibroblasts. So what are these fibroblasts? Fibroblasts are the very, very foundation of the niche of the tumor. They're non-epithelial, they're non-vasculature, and they basically form a connective tissue. And they make up the overall extracellular matrix and the bedding that the tumor is founded upon. So they're very, very important, and they're kind of like the first step that we have in tumor progenesis. So fibroblasts typically in normal settings are heavily involved in wound healing. And they produce a lot of transformative growth factors such as TGF-beta and PDGF. And this basically, excuse me? I'm sorry, was there a question? Sorry. So this basically leads to the activation of normal fibroblasts becoming the myofibroblast state. So what the tumor sort of does in the context of the microenvironment for to be able to grow is basically it treats this microenvironment as a wound that never heals, so that this myofibroblast state keeps on continuing, and we're getting a lot of growth factors and a lot of angiogenesis and blood vessels kind of coming into the area, allowing for the tumor to grow. Because if you think about wound healing, we need a lot of kind of growth factors for a wound to be able to come back to its normal tissue. So the cancer basically harnesses this power within the body and just uses it to its own advantage to keep on growing and to keep on getting a lot of blood flow and directing that at itself. So as we talked about, they cause angiogenesis, remodeling, and the epithelial proliferation. So this would be, I don't know if you guys can see my mouse. I think I would have to do this.
We can see it.
We can say, okay, perfect. So this would be a typical fibroblast that you would have, and then within the within the activation that you would have with the growth factors, this is what a myofibroblast and an activated fibroblast would then resemble. So more specifically, if we want to look at the role of fibroblasts in the tumor microenvironment, we can see, so this is a kind of like a brief overview of everything that they're involved in. So they drive a lot of the cancer cell proliferation by kind of the produced different growth factors that they have. What they do that's very, very interesting is that they induce more carcinogenic cell types. So they basically remodel the extracellular matrix, and through that, they end up remodeling the cancer cells phenotypically, and then the tumor results in a more aggressive variant. They also maintain a lot of the cancer stemness because they are involved in the WNT signaling pathway. So it kind of keeps the cancer cell at a mid differentiation state where it can harness the ability of extreme kind of high proliferative states to then grow. This is also a look at all of the other things that it does, such as driving angiogenesis. This one is actually extremely key because the more blood flow that we get to the tumor site, then the more the tumor has a chance to grow. So the CAFs will recruit a lot of endothelial progenitors, and they increase kind of vascularization within the tumor, and they also release a lot of growth factors, which then also in turn helps the vascular development within the tumor site. They also promote more invasion and metastasis because they cause tissue stiffness, and which is associated with progression to invasive disease. And then their exosomes maintain cellular communications with one another, which then also increases vascular permeability, allowing the tumor cells to now escape and travel from the initial tumor site to the circulation, and then have the ability to kind of spread out throughout the body, and then go on to also cause metastasis. So the second main tumor microenvironment cells that we have are epithelial cells that undergo epithelial to mesenchymal transition, or EMT. This is something that you guys might have heard. This is something that we do kind of talk about a lot within the context of HGSCA 3.0 ocular. So EMT is basically a process in which a tumor cell loses some of the epithelial markers that it has that are very much required for cell-to-cell adhesion, because that's what epithelial cells are great at, right? So the connection that they have to one another to be able to, for example, form the skin barrier. So they lose that, and then they kind of require more mesenchymal properties. So mesenchymal cells are very motile, right? So then motility and invasiveness then becomes now associated with the tumor cell, like Vimentin, which is what we use in our assay. So TNF-alpha and IL-1 beta are secreted by tumor cells and are some of the factors that really play into this EMT, epithelial to mesenchymal transition, and therefore cause the invasiveness that these cancer cells have, and can harness to cause then metastasis. EMT is very strongly linked to tumor aggressiveness and metastasis, and is basically necessary for the cancer to be able to spread. And this here is a look at what EMT looks like. So this would be kind of the phenotypic model that you would expect to see, with epithelial cells very structured and very closely knit. The cell-to-cell adhesion, it's very tight, and then you have, once they undergo the EMT, you have sort of a more motile state, something that can kind of be more invasive and easier to travel through the circulation. So this is sort of the epithelial cells in the TME. So this is sort of, if you're seeing on the right, we have the epithelial mesenchymal transition, and we have the red now showing vimentin. And what this does, as we went over, it improves the migratory and invasive properties, so a lot of the cellular motility, and it also increases tumorigenicity as a whole, because it's involved in double-strand DNA repair and induction of anti-apoptotic and pro-survival phenotypes within the cell. So the cells then live longer and are more resilient to receiving apoptotic signals, and then expression of pro-inflammatory and immunosuppressive cytokines, which is very important, because then again we're seeing that interplay. The more pro-inflammatory phenotype we have within the context of the microenvironment, the more the cancer begins to resemble the wound which does not heal. So then can harness the body's ability to send a lot of growth factors to the microenvironment. And then last but certainly not least, we have immune cells. So immune cells within the context of the tumor microenvironment, of course, play a very, very important role. The 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 only way for the cancer to progress and to survive and to even initiate is that it must have a way of evading immune responses or suppressing them. So we're going to kind of go over the general mechanism that it does so. So it has been known that tumor associated immune cells possess tumor antagonizing and tumor promoting sort of functions, and then they tend to target the cancer cells in early stage, and then the cancer cells sort of go on to escape immune surveillance and kind of have a lot of immune suppressive cells then kind of recruit a lot of immunosuppressive cells to the microenvironment, and then they inhibit a lot of cytotoxic functions that a typical tumor cells would have. So this is, I think, something you guys have sort of seen before, this balance of tumor immunity. So immunity as a whole is a balance between effector and suppressor cells. And suppressor cells are very, very necessary in a general context without cancer, because you don't want to have too much killing, because then that too much killing can cause a lot of immune disorders, and the cells can then go on to attack the body's own cells, and that's how you get autoimmune diseases. So the immune suppressor cells are necessary, and as are the effector cells, and there is a nice balance. But what the tumor does is, as it goes on to progress, it sort of tilts the balance, where most of, or almost all of the response that now you're getting from the immune system is mainly the suppressors, and you don't really have the effectors playing a role anymore. So this is a general um, overview that I wanted to show of the cells within the context of immunity. So we have sort of our innate immune cells, which provide kind of the first line of defense, whether it be against cancer, whether it be against even pollens that um, a lot of people are allergic to, and the body kind of responds to that. And then you have the most kind of sophisticated players, which are the Bs and the T cells that go on to produce your adaptive immunity. So within this array of all of these different cell types, you have some that can play different suppressor roles, as they can do kind of as well as activator roles. This is true, for example, for macrophages. Macrophages can suppress most of the immune response as well as are necessary to activate it. Same with T cells. T cells are the biggest activators and biggest effectors as well as the biggest suppressors. So based on the different kind of cell types that these cells can have throughout their differentiation period, they can kind of play for both sides. So this sort of looks at the immune cells now within the context of the TME. So we have the cytotoxic immune cells in both innate and adaptive immune response that can suppress the growth of the tumor by activating target markers that the tumor expresses, right? So some of the tumor suppressing cells that we're seeing are our natural killers, our M1 macrophages, our CD8 and CDT, so our cytotoxic and memory T cells, our dendritic cells, our B cells that go on to produce antibodies. And then the regulatory immune cells, sort of our suppressors, are part of the negative feedback loop that actively goes on to downregulate the effectors and also produce a lot of cytokines, which will, again, in another way, kind of lead to the differentiation of more regulatory cells as opposed to effector cells when the immune cells are now getting generated and proliferated. So that we can see on this side, and they're part of the negative feedback loop to keep basically the homeostasis and then regulate the immune functions. But because of the unfair tilt of the balance within the tumor, they end up promoting tumor growth by blocking the access of these effector and cytotoxic cells to the cancer cells. So this is a very general concept that's also very famous, which is the immune cells in the tumor microenvironment, the idea of the cold tumors and the hot tumors. I'm not going to go over this a lot because you guys have seen it before, but this basically talks about the kind of immune populations that you're seeing within the microenvironment. If you're seeing a microenvironment that is heavily kind of that heavily contains suppressor cells and doesn't have a lot of effector infiltration, such as activated T. You don't have a lot of immune exhaustion because you don't have that activation to begin with. So the cells never actually get a chance to get exhausted. And a lot of angiogenesis, that is a cold tumor. So not a lot of immune activity going on. And then you get your hot tumor, and a hot tumor that is a tumor that is now actively being targeted by the immune response. So this is an example of what the role that the TME plays also for therapeutics, because this distinction is very necessary when choosing what kind of treatment a patient receives, especially if that treatment is an immunotherapy. There is a high level of heterogeneity within the cellular subtypes of the TME, and it definitely has the potential to illustrate the stage of the tumor, the stage of kind of also the treatment, and whether or not we're getting the desired response that we're expecting from the treatment as well. And a comprehensive picture of a patient's tumor cells and TME together can definitely have a lot of implications for diagnostics as well as different therapeutics. And with that, I'll thank you all for listening.
So any questions for Sonia?
So Sonia, I know you're working on like an oncosome piece right now. Where does that really factor in terms of like this whole microenvironment piece?
That's a great question. So oncosomes are kind of they're known to be the different kind of communicators, right, between the cancer and its microenvironment and then the cancer cells together. They could be involved in niche selection for metastasis. So it does definitely can contribute to the microenvironment, but a lot more studies are needed on that front. So they're not as established of players. For example, fibroblast exosomes are, but large extracellular vesicles or oncosomes are not as well characterized as other factors within the microenvironment just yet, though. We'll see.
Anyone else? Maybe like we can also ask like have general question sessions after my talk.
Yeah.
Okay. So I'm gonna tell you some stories. Not necessarily all from our lab. Actually, nothing from our lab. It's more from the literature. So can you see my slides?
Yes.
Sure. Okay. So it's tumor microenvironment and IMC. So this is the overview of my talk. I will a little bit talk about tumor killing versus tumor suppressive microenvironment, imaging mass cytometry, and how can study tumor microenvironment with IMC and some future prospects. So what you see here is a mouse brain. And these authors, Bauma et al., what they did is they actually injected some human glioblastoma U87 cells directly into the mouse brain. And that's what you see. Like this is the imaging, which is also called as optical frequency domain imaging. And this is also in 2009. So what you see here is, this is the architecture of mouse, blood vessels and related like small capillaries that you can see. And here what you have is UAT7 cells which are directly injected in the brain. So, well, this looks very different than this one. So why? Because here tumor is growing and they actually build their own microenvironment. You don't see microenvironment much here, but that's 2009. What you see here is yellow. This is actually a 2 millimeter section or 2 millimeter thickness. You can see whatever is yellow is actually superficial, and the deeper part is in the red. So what you can see clearly here is this is definitely different than the normal architecture of mouse brain. You might read this article because this is very interesting to see how people were studying human microenvironment back then. And this is like 25 years study of human microenvironment, and this is 2009, so it's not late. It's just 10 years back. Now, as Sonia mentioned, there is a hot and cold tumor. I just want to briefly show how it looks like. So what you have here is biopsy and resection. On the left, you see cold tumor. On the right, you see hot tumor. What you see difference between two images is plenty of speckles here, and almost there is nothing here. So you have different markers which are in different color here: CD68 in macrophage, CD8 T cytotoxic cell, CD4 T helper cells. Then you have CD8 positive FOXP3, CD4 FOXP3 cells. So those are basically all killing cells. And then the red part is the tumor, and green part is stroma. So you can see here like stromal cells and tumor cells, they're always interacting to each other. And this is the overall density of tumor cells in cold tumor, and this is in the hot tumor. So if you have more and more cells, it's called as hot tumor, and then probably patient would have a better prognosis in this case. So again, this is in 2012. We had some antibodies and a lot of technologies coming in, but still there was a lot of things yet to be done at that time. Now this is the last year paper, and this is also what Sonia also showed in a table, and you can see difference between tumor killing immune microenvironment and immune suppressor microenvironment. Now what you have here is different kinds of cells, natural killer cells, M1 macrophage, then neutrophils, and as well as dendritic cells. And what you see is, so immune cells secrete something and tumor cells secrete something. So all these tumor killing immune microenvironment, what they are secreting here is all these perforins, phagocytosis, granzymes. So basically they are shooting all the bullets and all the fire so that this tumor cells would be, dead or maybe, under control at some point. Things are totally different on the right hand side. What you see here is immune suppressive microenvironment where you have T regulatory cells. They express certain different kind of markers, and also you have different M2 kind of macrophage instead of M1. Then you have N2 neutrophils, and you have a lot of MDSCs. And so basically, they try to keep themselves happy, each other's very happy. So this is also called as immune suppressive environment. Actually, a lot of immune cells are also helping tumor cells to grow or not to be killed by even drugs. So that's the biggest problem in kind of cancer treatment right now, is if your human microenvironment is supportive of the tumor, then it is very difficult to get the things under control. Now I'm coming to the IMC. What you see here is the image from imaging mass cytometry. It's a breast cancer biopsy. What you see in the red is basically pancytokeratin or pankeratin, and then in green you see smooth muscle actin, which is also a marker of stroma. Pankeratin you can assume is a marker of cancer, and you have DNA in blue. So why we need to study the microenvironment? Many of these cells, those also Sonia mentioned, are actually found to be prognostic in cancer research. So if you see macrophages, M1 macrophages or M2 macrophages, there are multiple papers showing that which are tumor supportive, which are actually good for prognosis. So then what stroma does is it also limits the access of therapeutic agents. When you are giving chemotherapy, some of the immune cells are also promote cancer cell survival, and distance from stroma can also affect cell state and differentiation. So in imaging mass cytometry, it is very interesting to understand is how far is your immune cells from actual cancer cells, and I will come back. I'll come to that. So you might have seen imaging mass cytometry lab. It's a small or maybe smallest room in our lab. It has two different units. So this one is called as it's also a site of one, which is also called as Helios, a commercial name. And the other one is Hyperion. That's the imaging system. So what it does is actually you can stain a slide with 40 different metal level antibodies. In traditional immunohistochemistry or immunofluorescence, you might stain the tissue or slides with like 4 to 5 different antibodies, because you have fluorescent label antibodies, then they have excitation and emission level wavelength, and there might be some overlap between the fluorophore. So maximum you can go 4 or 5. That's what our HDSCA does. We have 4 different colors. If you go use flow cytometry, you might see 10 or 15 or 12 different colors, but there's also a lot of overlap. But this one is a better one, where you can see the resolution is almost 1 micron and there is no overlap. So what it does is you ablate the slide, you visualize the slide, and you actually can reconstruct the pseudo image. And this one was initially designed for the tissue sections, but in our lab, we have also adapted it for the circulating tumor cells. So this technology was meant for tissue slides, but in our lab, we can also use it for single cells or circulating tumor cells or immune cell characterization. So how it works. Now, this is a grid of, let's say, 400 by 400 micron size. You have a tumor cell, and this I'm actually showing you for a single cell or CTC. What do you have? Like in HDSCA, you see a single cells in the middle, and then they are surrounded by a lot of blood cells. You can start with your laser, and it goes like 1 micron every time, and it ablates this entire section, which is 400 by 400 micron in, let's say, 20 minutes. So how it works? So you put your slide, in a IMC slide holder. Then you actually find the area which you want to ablate. Let's say this is the area you want to ablate. And then you have on top is a solid state laser. So, and this is your sample cone. So you actually target the area to the sample cone. So this is also a very high pressure under helium. So it starts ablating, and then whatever is ablated is basically ionized. And it's also called as plume. So this is actually taken inside this sample cone, and it's entered in the small tube, and then it is actually transferred to the site off, which is the other side of the IMC that I showed in the previous picture. So what you see here is every time laser ablates, the small square is 1 micron. That is the information you can get. Like every time when it ablates, it creates the image of whatever metals are present in that particular area. So what you have is, since you have 40 channels, you actually can get image of 40 different metals or 40 different antibodies. That means 40 different markers. So once you finish the ablation, basically you can stack all these 40 channels or 40 antibodies together, and you get a one image, which is basically you can use for analysis. Now, how do we use this one for microenvironment or like tissue biopsies? So just I'm gonna go a little bit in the basic is flow chart of immunohistochemistry. So you can take tissue sample or resection from surgery. Then you put this one in formaldehyde, 4% formaldehyde, just to fix the architecture of your tissue. And then what you do is you actually put this small biopsy in a cassette. So these are basically cassettes. And there is a machine called as a microtome. So what it does is it actually takes a small section. So this is your cassette. What you have here is very small thin sections of this tissue. That could be 4 micron, 5 micron, or 100 micron, depends on which assay you want to do with this section. So typically for imaging mass cytometry, what we do is we take 4 to 5 micron sections. Now, I don't put this another slide here, but basically you cut this open and you really have to fish it. What I mean by that is actually you take a single section on a slide, and these sections are actually floating in the water around 50 degrees Celsius, and then you put one section on each slide. So once you cut this, probably you might cut like 20 sections, and you might have like 20 slides out of it if you don't lose anything. So this is basically what biopsy slides that we get is all has been processed by this way. Now, since you have the slide here, this is same as immunohistochemistry. The only difference is instead of putting the primary and secondary antibodies, now you're actually putting the cocktail of antibodies. So what we do in IMC is we have 30 to 35 antibodies in one single experiment. These antibodies are actually conjugated with metal ions, and these metal ions are lanthanide rare metal ions, which are actually not found in human body. So once it conjugated, you have a stock of those antibodies. You decide which antibodies you want to use for your study, and you make a mixture of it. You decide different concentration or dilution based on your assay, and then you basically you use that cocktail of antibody to stain the slide. And then this is the Hyperion. So you put the slide here, you ablate with the laser. That's what I showed in previous slides. And then the information basically transferred a small tubing in between these two instruments. And this is the basically one which reads everything. So this will give you pseudo image which you can analyze in a further different downstream platforms. So in immunohistochemistry, what you see is one single brown color, because you can use only one color at a time, or maybe 2 to 3 colors if you want to use consecutive bleaching and restaining procedure. You might use the same slide 2-3 times. You might see 3 different markers. But here in a one single experiment, you can get an image of 30 to 35 different antibodies or protein markers. So how it is used in microenvironment study. So I'm going to talk about 3 different articles which are published in recent time because it's just going into background. Imaging mass cytometry technology is quite new. The machine was commercialized a little bit like 3 years back. CyTOF was commercialized 10 to 15 years back. So whatever the results that we are getting with IMC regarding tumor microenvironment is actually all are recent, 1 or 2 year old. So this is a paper by David Rim. He's one of the famous pathologists. You should see his talks on YouTube about how to use different antibodies in different assay, and even he will talk about how different antibodies, which are actually therapeutic antibodies, are not also a very good antibody. So it's a very interesting talk. You should listen to him. So in this article, which is multiplexed 18-plex measurement of signaling targets and cytotoxic T cell in trastuzumab related treated patients using imaging mass cytometry. So they had like 18 different antibodies they used. The first thing that they have to show, since it's a new technology, is it works exactly like your gold standard immunofluorescence looks like. So they showed that, okay, it's exactly like what you see in IF. And what they see is, if there are a lot of cytotoxic T cells in the tumor, it's more prognostic. And they also claim that, they also actually conclude that if it is a lot of cytotoxic T cells, it's a complete response after treatment with trastuzumab, lapatinib, and combination therapy. So now people are using more and more imaging mass cytometry to actually see different markers and see like which protein markers are more prognostic and can be used in the clinic. So mind that imaging mass cytometry, it's still not officially part of any clinical trials because it's not CLIA certified technology yet. It's not. It's only for the research purpose. It's not like a diagnostic or something. So since a lot of papers are coming up, I'm hoping that in coming years, this would be also a part of clinical trials. So currently, CyTOF or your traditional flow cytometry is definitely a part of clinical trials. This is the paper, which is a landmark paper by Boden-Miller lab. And if you're interested in IMC, you should actually, it's not here, but you should actually look at Bernd Boden-Miller from Zurich, Switzerland. His papers in imaging mass cytometry, and Jackson, his first name, yeah. So he was its postdoc, and now he has started his own lab in Canada. So traditionally, what you know in breast cancer are like, actually, you have hormone receptor positive cancer, or HER2 positive cancer, or basically triple negative cancers, right? So all the treatments or everything is based on these 3. Now what they have done is, here they actually did the imaging mass cytometry with around 352 patients. They used 35 different markers labeled with metal ions, and they had like hundreds of images of single cell resolution. And with that, what they found is there is not like 3 or 4 different kind of markers which could be prognostic, but there are population of like I think 18 different population that they found which could be prognostic. I'm not going into the detail of all these images, but these are basically our Kaplan-Meier curves. On y-axis, you have a percentage survival, like 100%, and then like 0%, and then here you have time or months survival. So whichever is like this, basically it's patients are dying more often, like prognosis is really bad. And if this is like, if it is like this, it's basically 100% of the patient survives. So what it says is, you can identify different kinds of populations based on these markers, or you can stratify the patients based on these markers, and can identify which patients would have more prognosis, better prognosis compared to the other patients based on these markers. And treatment can be actually, stratify according to that. So this is the first paper, probably where they have like more than 300 patients, and you can actually can do that. And this is not like typically only for ER positive or HER2 positive. Basically, they did all breast cancer. So this is like a bunch of everything. And now like you can go, let's say in only triple negative, and you can still see what different markers are expressed. So you can actually go in details of each kind of breast cancer subtypes and see what different tumor population, what different immune population really contribute in patient prognosis. And this is a little bit outside of the cancer immunology. This is in COVID-19. So this technology can be used in COVID patients. And this is one of the first paper actually was published to see how immune cell infiltrate into the lung of patients which are infected with the COVID-19. So on top, you see a control patients, maybe a lung sample from a deceased person who has donated their lung. So you don't see anything basically. So there are not a lot of immune cells. So that's what I'm gonna say here. Patient one, I think he is a 94-year-old person it's here. 94-year-old person who died of cardiac arrest, but also was COVID-19 positive. And patient two was 65-year-old who also had COVID-19 and then also had a pneumonia complication. So what you see here is different markers like CD3, these are all T cells, CD3 plus CD8 is basically T cells, T cytotoxic cells, and this is T helper cells. And you have CD45RO is like T memory cells, and CD16, I don't know what is CD16. But what you can see here is there's a lot of infiltration of CD45RO in this patient. And you would see that immune cell profiling between these two is very different as well. It's very different compared to the control patient or a healthy individual. So definitely immune cell infiltration can be seen with IMC. This is the power of this technology. You're not really looking at only one marker or two marker or three marker at a time, but you're actually looking at plenty of markers. And then here they just talk about CD45RA, but you might find plenty of other markers or other immune cells which might be overexpressed in the COVID-19 patients. So in all, basically what you can see is this is one of the awesome technology developed with the time. So we started with 2009 where you can have imaging, then you have a lot of flow cytometry, then all those markers that Sonia also mentioned. Basically, people have studied using flow cytometry and cytop, which is also a suspension mode of imaging mass cytometry. And then now, since last 3 years, we can use IMC, which is a tissue imaging. So what you can do in that is you don't lose spatial information. So when you use flow cytometry or suspension cytop, you get a biopsy, you basically crush in single cells, then you have single cell suspension, you stain it and you run it. What you do is you basically can see all the markers, but what you have lost is a 3D architecture, or you have lost the spatial information, which is the power of imaging mass cytometry that you can study and you can look into it. Okay, so coming some future prospects of this technology, and I would start with a very low hanging fruit at this point and not published yet, but I'm sure some good paper would come in coming years. What is that? So I mentioned like how you make those tissue slides. So you can study consecutive sections of tumor biopsy. Like let's say you have 4 sections, 4 micron each or 5 micron each, then basically you have 5 into 4, like 20 micron box or square, where you have all the cells. Maximum size of cell is like 15 micron, immune cells, big cells as well, big tumor cells. So you basically stain those 4 consecutive slides, and then you actually overlay those 4 images. What you get is 3-dimensional image of 30 markers or 40 markers which are expressed in that small biopsy or cut. So you can stain immune cells, you can stain tumor cells, basically 30 markers, 4 sections, consecutive, a lot of information. you can actually make a movie of it, like what are different markers are expressed there, how they're interacting, and what we can learn from that. So that definitely is coming in coming days. What we need to do that is data analysis. So we need to have a better tools to segment the cells, because you need to have a single cell, boundary of single cell, where it ends, where it starts, and there is a lot of overlapping at this point. So how you can segment the cells in a better way, and how can you analyze the data using different methods. Right now there is no such methods available. There is Histocat, which is again prepared by Boromiral Lab. It's open source platform, and there are a couple of commercially available. They are very expensive and not that trustworthy. So if you are interested in data science or making those nice tools, this is the area probably you can also consider. Third is, obviously we need a lot of antibodies, and we need to build those panels. We need to conjugate this antibody, validate that antibody, also do some assay to find right dilutions. You have to titrate that and build those panels which actually can be used for assay. So that could be in tissue microenvironment, or that could be also in our single cell immune cell profiling and everything. So having good antibodies really, really, really important. So that's all from my side. So if we have like some questions, I think we still have 5 minutes to discuss about in general tissue microenvironment, or any other questions if you have. Thank you.
Students, no questions for our presenters? Thoughts, ideas, comments?
I know just from my very brief times where I've gotten to see some of the IMC images that we've produced, the resolution is, there's a very clear cutoff for the resolution, whereas the slides that are processed with HDSCA, they aren't as pixelated, I guess you could say. Is there a way to improve that resolution, or are we limited physically by the size of the laser that we can use?
Okay, it's a very good question. So right now the resolution is 1 micron. So I think it's not as good as a 40x magnification that you get with your microscope. Not definitely with the confocal microscope. Unfortunately, the resolution is not something. Yeah, they could not change that yet. So it's a lot of physics involved in that. So I cannot give you a technical answer to it. But yeah, so right now 1 micron is the resolution, and that's also one of the limitation of this technology.
Okay.
I had a question about the microenvironment. So I know that tumor hot environments kind of respond more to immunotherapy. So what are the current methods to turn kind of immune cold tumors to immune hot tumors?
Sonia, maybe you can answer this better than me.
Yeah, that's a great question. So the very famous Nobel-winning experiment of PD-1 inhibition therapy and PD-1 and PD-L1, that is one method. That is one thing that they can target. Because for example, there would be a lot of PD-1 within the tumor cold environment, and then if you try to kind of inhibit that, there are other markers within immune therapy too, like CTLA4 and different markers that you can do that too. But the most famous is the PD-1 PD-L1 dynamic. That if you can target that, that can potentially make an immune cold tumor receptive to immune therapy.
Anything else?
So Milind, when you're building a cancer-specific, like a specific cancer type IMC panel, what are the most important elements to consider in that process?
So this is for our slides or for biopsy?
We could say for our slides.
Okay. So very first thing is, there are plenty of things. So you first start with what our HDSCA is. HDSCA is immunofluorescence staining, basically. So you need to find antibodies that are actually compatible with that technology. So immunofluorescence, immunocytochemistry, immunohistochemistry, those antibody would work. Don't use antibody which are made for flow. Those not gonna work.
Yeah.
So that's the first thing. Second is we use mouse antibodies in our HDSCA and secondary antibodies as well. So basically goat anti-mouse. Then again, now you use mouse antibody in IMC, those will bind to your secondaries from HDSCA and you'll have a non-specific signaling, non-specific binding, sorry. And you will see lot of markers are positive. So that's not true actually, because that's basically non-specific binding. So get rid of all mouse antibodies as much as possible, or at least test if they are not showing you any non-specific binding. So what we do that for is we use all rabbit antibodies, and then you need to find antibody which are BSA free, because a lot of antibodies they actually sold in BSA because that's basically stabilization buffer. So when you conjugate that with a metal, your antibody has to fight with BSA, like who's gonna go conjugated with metal. So BSA is since dominant, that will conjugate it and your antibody will be just out. So you don't want to have an antibody with BSA. So that's one thing. You get rid of all BSA, or you just buy antibody which is only in PBS but not in BSA. So these are 3 or 4 actually important things that you need to start with. And then you need to validate those antibodies with positive and negative control slides, which are exactly processed like how we process with HDSA slides. And if we have specific signal at specific place, like let's say you're working for antibody which is expressed in membrane, and you see that in membrane in positive control slides cells, not in negative control cells, then that antibody is validated. So with that, you actually build a panel. So last few years we contributed antibody, and we have now around 60 antibodies which are compatible with HDSCA.